So I'd like to welcome you all back to the afternoon session. We have three back-to-back speakers. The first one is Lisa Getzendaner. So Lisa graduated from Duke University, the PA program, in 1992. And in 1993, she started working at Fred Hutchinson. And then in 1995, she transitioned to the Unrelated donor program at Fred Hutchinson. And she has been the clinical liaison between the search teams, the referring physicians, the donor registries, the transplant teams, and the patients for 20 years now. She is the go-to person for searches. So she's going to talk to you about HLA typing, donor selection, and beyond.
So you guys came. Thank you. I was wondering if I should come. No. So I have 25 minutes for a really big topic, and Karina knows I love search. I love HLA typing. I'm one of those really weird people, and I'm going to try very hard not to put you to sleep with too much HLA mumbo jumbo, but at the same time, I think one of the big problems we have in our general clinical community in transplant is most of our general transplanters just leave the HLA to the HLA guys and then wonder why we can't find what they want. So I'm hoping to impart enough information to you here today that you can understand why easy searches are easy, why hard searches are hard, and then the pitfalls that your search teams will fall into. So how many people here actually look at registry reports and actually do searches? So a few, but not the majority. Okay, so good. So I'm not going to probably tell you guys too much that you don't know already. But so today in my talk, I want to spend a little time going over function and structure of the HLA complex, spend some time talking about genetics. We'll spend also a bit of time talking about typing, how we type, the nomenclature of HLA, which has gotten incredibly complex since I started in the alphabet soup we live with today. Spend some time talking about how you look at donor searches, both from family donor perspective and unrelated perspective. We'll talk a little bit about HLA typing and transplant outcomes, and I'll give you some insight as to how we put our search paradigm together in Seattle, with the understanding that every transplant team needs to do the kind of transplants they do best, and thus how you structure your search is going to depend on your research priorities and what you do well at your center. Oops, wrong way. Dang, I knew I was going to do that. So, basics of HLA. What is HLA? Human leukocyte antigens. I actually talked to a fellow group and I said, somebody tell me what HLA means, and they all looked at each other. It's kind of funny. Keep in mind that HLA, the class 1 and class 2 genes are expressed differently in different cells in the body. So class 1 is pretty much ubiquitously expressed, whereas class 2 is much more tissue group specific and it's inducible. And keep in mind, this is our immune system's way of telling self, of telling good from bad, of telling infected or cancerous from non-cancerous. And it's, really the basis of how our immune system functions. And I'm doing it again. All right, I'll get this right. So, what are we looking at when we type for HLA? So the class 1 proteins, the A and B and C, are created from a single chain with 2 alpha helices that create your binding groove. So that binding groove that sits at the top there, that's where the money is. So that's where the immune system can then present antigen and cause the cascade and reactions that we see. And class 1 and class 2 is structured a little bit differently, and because of that, they can do things a bit differently. That's a whole lecture in and of itself, and frankly, beyond my day-to-day working knowledge. So just know that they're different, and know that they're structured different, And because of that, when we type, we're looking at different genes that create those proteins. So keep in mind, HLA is really simple genetics. It goes right back to high school and Mendel's P's. It's all coming off of chromosome, the short arm of chromosome number 6. But also keep in mind that HLA genes are grouped together. You have the class 2 at one end of the complex. You have the class 1 at the other end of the complex. And because of that, these genes tend to travel together. And we're going to talk more about that in a few minutes. And recombination events tend to happen in particular ways. You see recombination between class 1 and class 2, which is what's given us incredible diversity in HLA haplotypes. And when I started doing HLA typing and such back in the early 90s, it was all done with serology. We had just a handful of recognized antigens. Outside of some DRB1 where we knew there was allelic variation, an A2 was an A2 was an A2. Not so much anymore, guys. So there's over 12,000 HLA alleles identified now. And they add probably 100 a month. So it's incredibly complex. Do you need to know them all? No. But there's a lot out there. Now, a lot of it are variations outside of these binding grooves. So from our perspective as transplanters, doesn't really matter. Stuff that really matters to us are the allelic variations that happen within the binding grooves. But for example, HLA-A1 right now has 235 different described alleles. So it's complex, and if you really like HLA and you read the HLA literature, the HLA immunologists and the guys that are really into immunogenetics will wax eloquently about why we do that. So again, going back to the polymorphisms in these genes, what we're really worried about is what's happening right up here in the groove. And so when we go and type for A, B, and C, we actually only really look at the second and third exons in those genes. And within those exon 2 and 3, then within that, it's the variations in these alpha helices that really make a difference. So most of the other variation is happening down here in the transmembrane sections of the cytoplasmic tails. Now with the class 2, you actually you only have to sequence one exon, exon 2. And again, within there, it's this variation right up here in the groove. That's where the money is. So it's important to know that your lab can define allelic variation if they sequence, and when we do complete sequencing, they'll give you exon 1 and exon 4 and exon 5, and you don't really care about that. It's okay. What your good HLA specialist in your lab should do is tell you, is there a functional variation between the two antigens or alleles you're looking at? And if you look at any standards for HLA typing, it'll tell you you sequence exon 2 and 3 for class 1, and you sequence exon 2 for class 2. And outside of that, right now, don't worry about it. But again, with the technology now that we're seeing in typing, they're going to give you way more data than you really need for matching, which comes around to nomenclature. So back in the day, an A2 was an A2 was an A2. Now they've added this lovely plethora of information, and you're actually not going to see this on HLA reports like this. What you will see is what you're looking at, an A, and what the antigen or allele family is, an A2, and then what allele you have, the specific allele. And so this happens to be an O2, O1, O1. I should have picked one a little more common, like an A201. You won't see these, the second, or the third and fourth. These are DNA substitutions happening in other places that really are non-coding, like the field 4 non-coding regions. The field 3 are substitutions that actually don't change the protein. You will, however, see these mainly two-letter suffixes coming off the end. If you see an N, that's a null allele, which means you can type it, but it doesn't generate a protein on the cell surface. And you also see L's, and those are low expressors. So those allelic variations, there's some protein on the surface. The nice thing about those kind of alleles is they're very tightly tied to haplotypes. So even in the unrelated donor setting, if you type a patient with a null allele on a haplotype, very likely you may be able to find an unrelated donor carrying that same type haplotype. So the common null alleles, the common low expressor alleles travel together with a haplotype, and you'll find them. You occasionally run across a really weird patient that has a true null allele that's not tightly linked with anything. We actually then will not search for that. We'll search for a homozygous donor and try and not match for the null allele. So we have a bunch of techniques, and when you're looking at old typing data, registry reports, you're going to see a large spectrum of results. And you have to look at those with a critical eye and know what techniques were being used to generate those typing reports for you. So old data will not have this asterisk. So if you see an asterisk, you know that's been done with a DNA technique. Old data will have an S, and that says that that's serologic typing. So be careful, because we missed a ton with serologic typing. Though common being common, serology typing of common haplotypes will generate at a DNA level what you want. So most all typing today is done with DNA-based more allele-specific data. Most of our data is generated on the registries and such with SSOP or SSP. The nice thing about these techniques, we've created primer sets that separate common. So we've got a primer set that will separate an A0201 from an O205. Those are the two most common A2 alleles. So you want a primer set that maybe won't sequence the whole gene there, but will give you the common differences. So we do direct sequencing of our patients so that we do know what HLA typing we're dealing with, and we do bring donors up to sequence level, but you don't necessarily need to do that on a search, and it's certainly still quite expensive to do that, even despite the progress being made to streamline this, mainstream it, and make it a lot more inexpensive. But keep in mind, you're going to see now the alphabet soup, because these primer groups give us the common, like an A2601, but then all this other stuff, if you sequence that loci, may show up. So we create, I don't know how often they bring out new codes, but I think it's on a near monthly basis, and it's scary. I send that all back to my search coordinators and say, please give me a number on this and let me know if I've got a match. There's also really nifty websites that you can just type in HLA allelic codes, and then usually you can just type your code in and it'll give you what your common alleles will be for that code. So just some basic terminology when we talk about search. So a haplotype is that linking of the A, B, C, D, R, and those genes travel together and are inherited together. When we talk about genotype, that's when we have a family study. So that's when we know which A, B, C, D, R came from mom, which one came from dad. Keep in mind that most folks are heterozygous, so you have 2 different A's, 2 different B's, but there is homozygosity that pops up, and that can have impacts for how you do donor selection. So prevalence, that's just how often you see a particular allele in a population, and keep in mind HLA is very, very tightly linked to your racial and ethnic background. And then we'll talk a little bit about linkage disequilibrium. So if you ever go to HLA talks, especially with the HLA gurus, they love linkage disequilibrium. And basically that just says these things like to travel together. And if you have a patient that has the wrong HLA antigen for a particular haplotype, you're going to fight LD in your donor search. So this is kind of what I'm talking about. So if you have an A1B8DR3 haplotype and the frequency in a particular population is 16, 9, and 10%, then if you just do basic math, you'd expect if you link these all together, that would actually be the frequency of seeing them together. But what we actually observe is that they're much more frequently linked than if you look at any one by itself. So that's the principle of LD, or linkage disequilibrium. So that is because you can see how close these genes are sitting together. So DR and DQ are very close together. So random recombinant events, which is how we get genetic diversity, don't tend to split those. Same thing down here with HLA B and C. HLA B antigens typically have 1 or 2 C antigens that travel with them. And that's, again, because they're very close together on the chromosome, and recombination rarely happens. Now you see you have a big split here between A and B, and then if you look across the whole complex here, it's quite easy to get recombination events between class 1 and class 2. So this is why linkage disequilibrium exists, and then it's also why you get in trouble if you have a bad antigen in a search. So going back to looking at related donor typing, again, back to Mendel and his P's, you get one from mom, one from dad, and that's your haplotype. I could have gotten fancy here and created a crossover event with a SIB that doesn't match anybody, but I didn't do that. So in any family study, if you're actually fortunate enough to have the money to do a family study and have enough people to do a family study, it's pretty easy then to sort out how the haplotypes exist. And in this case, if this is our patient, we have a matched sibling. Now, if this SIB didn't exist, we'd have a bunch of SIBs that nobody's matched. And if you've been to some of the earlier talks today, there's all kinds of fancy stuff going on that if you're going to pick a haplodonor, maybe you want to pick a haplodonor that has the non-shared maternal haplotype, or the NEMA. And there's all kinds of little games you can play if you actually have a family study and know how the antigens have been inherited. And again, common things being common, with very common haplotypes, like I'm showing here, the 183 is almost always going to come together. That's the most common Caucasian haplotype there is, and they tend to travel together. But without a family study, you can't prove that. And Dr. Petersdorf at the Hutch did some really elegant studies a few years back that actually showed that there are many different ways to get your HLA complexes. So she did look, this was mainly done in unrelated donor transplant, where she looked at what haplotypes the patient had And although in this setting, this patient is phenotypically identical with all these donors, they did not actually, aside from this donor here, inherit the haplotypes. So one could imagine if you inherit the same haplotype, all that stuff in between the 5 major genes we're looking at will likely also be matched because, again, common things travel together. Outcome data from this study showed that if you actually had a haplotype matched donor, transplant outcomes were better, transplant-related mortality was less. To do this typing is incredibly difficult and certainly not something we can routinely do. But it's an interesting idea that we're seeing in cord blood transplantation because we actually do have a lot of haplotype data in the cord setting because we have maternal typing on a lot of these units. And again, polymorphisms that you see will exist on unique haplotypes. So if you have a really odd A1 allele and you're fighting an A1B8DR3 haplotype, it's hard to find that in the unrelated setting if you have something very unique because it's probably a unique haplotype. So matching. So we have antigen and allele matching, and I spend a lot of time answering questions about, is this a matched transplant? So when we talk about antigen level matching, we're just looking at the first digits. An A2 is an A2 is an A2. And then you look at the second digits, and yeah, it's an A201. This is both an antigen and an allele match. This setting here, you're actually mismatching at the allele level. Antigen levels are matched. You both have A2s, but you have 2 different versions of A2. And I use my green apple and red apple analogy when I'm talking to patients about that. Then you get into the full antigen matched setting where you've got an A1 versus an A2 here in this setting. The data now is really saying, you know, you can delude yourself and think that this is a better match, but it's probably not a better mismatch than this one, but that's probably no longer correct as the data sets get larger. The other thing we have to look at sometimes is this question of homozygosity. So keep in mind that when we mismatch, as in this setting here where we're mismatching at a heterozygous loci, so 2 unique antigens at the site, you have both a rejection or a host versus graft vector and a graft versus host vector. So great, fine, it goes both ways, happy days. There's new data coming out, though, that for a long time, this idea of mismatching that creates only that graft rejection vector was kind of scary because old data in the CML age, these grafts we lost, and we had much higher graft failure rate. And that's probably because we had untreated patients, we weren't immune suppressing very well, and we probably didn't have as good a matching going on as we hoped. And so in Seattle, we actually excluded this kind of mismatch for a number of years. But there's some very nice data coming now out of the CIBMTR that shows that this mismatch is actually not so bad at all and probably better than this mismatch here where you only have a GVHD vector. So the nice thing here, for the purposes of GVHD, this is a matched transplant. Now, you can get into cure, you can get into a whole bunch of other stuff that maybe overrides that some, but from a practical standpoint, there's pretty good data now saying that if you are going to mismatch at a homozygous loci, it's kind of nice to do it in this manner. Be careful of your protocols, though, because a lot of the protocols still say you can't do that. So when we're looking at actually typing and donor selection, we've got patient and sibling typing here. It's usually at a lower resolution. I start calling the lab right off the bat. I don't care if the DRB1 alleles aren't out yet. What do we got? And you pretty readily can find out, at least out of our lab, within about 5-6 days, if they can look and say, I got something good or not. And if you've got a couple siblings, you can even start telling your haplotypes pretty quick. So in this first example, do we have an HLA match donor? We do. The patient does match the second sibling here. And if you want to ask the haplotype question, you actually can answer that here because this other sibling has very nicely presented the other two haplotypes. So you pretty much have all the information you need from this family study. This example down here, where you only have one sibling donor, oh yeah, there you go. So they're not matched. And on top of that, when my docs start calling and saying, okay, what's the world book look like? Do we have an unrelated donor? I really can't say much of anything because I don't have high enough resolution typing to run an effective world book because I don't know what these DRB1 alleles look like. So it's pretty easy to say when you don't have a sibling donor. It's a little bit harder to say when you do. And this is back to the alphabet soup world. This is what we deal with day in and day out when we're looking at registry reports. So do we have an HLA identical unrelated donor? And the answer would be yes. There's this one right here. And if you do the alphabet soup, they match. But likely, all of these donors are matched. You're just seeing a bunch of donors being presented with a bunch of different levels of typing. And common being common, they should have the common antigens as well. And I wouldn't go to workup with this donor here, but I would go to workup with any of these donors and then bring the resolution up. Oops, I went the wrong way. So we do have very nice data coming out of registry studies that say if you have a matched sibling donor, use your match sib. From a pragmatic, practical perspective, I'd hold to that. Now, we heard data today or through this week about older sibling donors maybe not giving us enough T cells and things like that. You know, it's pretty hard to pass over a readily available donor sitting there in your midst. Now, we do know that the SIB donors back in the day of low-risk disease really did better than the unrelated. This is old data, though, and Dr. Wolfrey updated that data with advanced disease, which really didn't show a survival benefit in the matched SIB setting. So the corollary to that is if you're willing to transplant with a SIB, you probably should be willing to transplant with a matched unrelated as well. But you have to balance the risk of the disease, how aggressive your disease is, and how worrisome the relapse risk is. Oh, I went the wrong way again. And then match grade is key. So this is what I was telling you. Don't be deluded to think an allele level mismatch is as good as an antigen, is better than an antigen mismatch. And Dr. Lee published this data in mainly lower risk marrow transplants. That was updated by Dr. Wolfrey that again showed that mismatching, even for a single allele, had an impact on mortality. So mismatching is bad, but if you don't have an option, it's probably better than not transplanting at all. But these are really the settings where your group needs to then look at what are you going to do in your new preparative regimen and your new GVHD prophylaxis to try and abrogate this increased risk that the mismatch brings along. So I would say we shouldn't just keep doing the same thing because we know it's not as good. And then we just had this very nice study come out with 8,000 donor-recipient pairs. And the take-home message to this is that all mismatches, A, B, C, all create risk. And the risks are actually fairly uniform. Some are a little bit higher. The relative risk for a mismatch is a little higher. I don't believe this DR data. It's a very small group. There's only 217 cases out of the whole 8,000, and that's because we quit doing these kinds of transplants a long time ago. So again, I don't believe that we can overlook a mismatch at DR yet. Oh, I'm running over. All right, so really quick, don't forget antibodies. If you're mismatching, you need to make sure your patient doesn't have a donor-directed antibody. That's really important. We've been long advocates, and there's a variety of ways to look for these antibodies. It's pretty easy to avoid mismatches where you have antibodies. And there's a lot of groups that have had problems with engraftment, and it's because they don't look at antibodies. And then, so cord blood selection is the other thing. Make sure you don't have antibodies against your mismatch. And we're doing a lot of work now in Seattle. We've kind of abandoned the 6 of 6 model. We're moving to 8 of 8 allele typing and combining that with CD34. And so we often are passing over 5 of 6s that are really on an 8 of 8 level, like a 3 of 8, and moving instead to allele level typing. There's beautiful data coming out, especially in the single cord setting, that this is really important. And it's actually amazing how easy it is to find with the high level of typing on the registries what that looks like. So I think at this point I'm going to stop, and I think we have a couple minutes for questions. Two questions. Yeah, I'll be in the back. So, questions? No? All right.